Okay, so let's start day 3 of the workshop on Cytoscape. We're gonna go over additions to Cytoscape to allow us to get data in from the online resources, and that's probably most of what we're gonna do today. And it's gonna be mostly clicking on Cytoscape and web browser. So we're out of PowerPoint. For now, back to Cytoscape. Okay, so to get interaction data easier way than using the default Scikit interface that we've been using yesterday and 2 days ago, we need some extra apps, extra modules that can be added to the default version of Cytoscape. To do this thing, we can go to apps, go to app manager, and we will need 3 things. We will need bingo. We will need intact app. Hopefully I do have no, so I've got to get it one at a time. So bingo, select it, install, and it should go in. And then we'll need intact, which is here, and the same protocol install. Sometimes when installing those things, you might get a message, different apps, you might get a message that you've got to agree to some sort of a license, or you might have to download it as a file and then install from file. So there might be variations, but in most of the cases you're gonna get additional modules installed by just selecting it and adding, as I'm doing it now. And the third one that we need is this ring up here, and then install it as well. Okay, so those are 3 things that we'll be using today. Okay, and if you look now at the search field in the panel here, you're gonna see that now we've got more things showing up. Initially, all you see is NDX and NDEX and Scikit. Those 2 things are default. We've added intact options and options coming through string. Okay, so let's start with our toy network, the simpler one, the Cytochrome BC. So I'm gonna get it in as before to have something to work with. So let's import network from file. I want our Cytochrome BC open, and as before, I don't want the first line to be treated as header, so checkbox goes off. First column is protein, third column is protein. On the other end of interaction, the middle one is interaction type. And we ignore column number 4. So, okay, and here's our network corresponding to Cytochrome BC1 from East. So that's what's here. Now, interactions came from the PDB file. So this is pre-made network that's based on one particular structure, but in principle, the proteins that you import can be coming from your experiments or from somewhere. Okay, it's wherever. Let's see, wherever Eli is getting. Oh, he's getting. Okay, so Eli is taking care of it. Okay, so how to get interactions just from the list of proteins? Well, we did try before the default psychic thing, okay, but we can try the other two ways that we try today. So to do this thing, we can always go to node table, then select the right click on a column to copy column values. Okay, and then we'll start with string protein query. Okay, and we can paste Control V, our identifiers in the field here, and then in the options here, we do have to specify species. If we try it as now with default Homo sapiens here, we can try to search, right? And there's gonna be something strange happening. Okay, let's try. I might have problems with, well, this should be Saccharomyces. So Saccharomyces. Here it goes. Yes, here we are, and try. Okay, and here we've got the network that was provided by string. Okay, depending on how many things we want to get apart from our query, we can adjust the confidence score and we can adjust the maximum additional interactors. With defaults, you're getting only the proteins that you queried for, and you're getting some pretty abstract cutoff of 0.4, which translates into a subset of interactions that string deems relevant.
Why is it letting me choose the species?
You should be able.
But when I click on it and try to type, I could try restarting.
Because you're supposed to go
I see.
yeah. I think for diseases, it's probably only human, so you've got no choice there. Okay, so this cutoff for confidence is pretty abstract, and to see how it works, we can go to actual string web page. Let's say here. Okay. This is the website of the string database, and the queries through Cytoscape are actually querying the database here. So this is more direct of looking for things here. And the advantage of using Cytoscape is that you can query for many proteins at once. On a web page, you've got to specify only one protein. So I'm going back here to node table and steal the first value here, and then I paste identifier. I'll pick up here.
I think
Mhm.
if you go to multiple, because I have used the website, and if you go to multiple proteins on the left,
Oh,
you can paste the
Oh.
other per.
okay. So this is another option, but then it's gonna be messy. So that's true that multiple things can go here, but if let's well, we can do it. Why not? Let's see. So then you select here, and then copy column values, and then paste here. Okay, and then organism author should be move it. Okay, here we want Saccharomyces. Okay, and then search. You is gonna now check if all those things are correct. Okay, continue. Okay, and that's what we're getting. This is equivalent of what was done here. Okay, but it's easier to see what's happening when we're wiggling different parameters. So first thing to realize is that connections between two proteins, let's say those two things here, are based on different types of information. Okay, and different line colors correspond to different types of data or predictions. And according to the legend here, what's possible is you might have data that's coming from different databases. Some of them might correspond to data that's coming from experiments, but not necessarily so. So there's distinction between created databases and experimentally determined interactions. Then you've got predictions based on predicted function that's similar between the two proteins. And then you might have other things, which is either text mining, which essentially means that names of the proteins co-occur in a paper. That's roughly what this text mining part is. Connections are made between proteins that were studied at the same time in one paper. So it is a hint that proteins do have something to do with each other, but it's not guaranteed that they interact with each other. There might be co-expression, and there might be a connection because proteins are homologous, which has also nothing to do with interaction. So this is something that one's got to be aware of. For one particular interaction, you can click on a link on the edge, and then you're gonna see where the evidence is coming from. Okay, and then each of those evidence types do have a score, and then there's some overall combined score that combines the values for different from different data sources, and then the cutoff that you specify when importing stuff into Cytoscape is essentially cutoff on this number. And this doesn't guarantee that you're going to get only physical interactions, even if the combined score is high. It's gonna vary from case to case, and it's hard to track. You can set the cutoff relatively high, somewhere at 0.6, 0.7, to see what's happening, but still there's no guarantee what got imported. And in contrast to the website, where you actually can see details, what's contributing to given connection. Okay, if you go into experimental, you can go to show and see actual information about the experiments or where they came from. Some of them came from Intact, some of them came from BBB, and so forth. In the network imported from into Cytoscape, you see just an edge, and if you look at annotations of those edges down here, okay, all you're gonna get are the scores provided by string corresponding to how reliable they deem the each contributing part of the edge is. Okay, so but there's no information about actual experiments. There's nothing there. So this is limitation of the import from the string going by the when using the app. It's just very top level information. Details are completely lost. It is also possible to get more than just the proteins we queried for. To do this thing, you can go to upstream and expand the network, okay, and then you can specify how many more proteins you want to get in. Let's say, let's ask for 50, okay. You've got a slider that decides or determines how those 50 proteins are selected from possibly more things connecting to proteins in initial network. But this is once more not transparent in a way that you can't specify you want only physical interactions or you want co-expression. There's no way to choose which source of data is coming from. So we can add another 50 nodes here. Okay, we will get bigger hairball. Okay, we can after importing we can use, let's say, organic layout to get stuff in. If it's too crowded, and then we can go and make it a little bit bigger. But we're essentially getting a hairball with a bunch of edges, and then if you look at the edge table, and then you're gonna see scores for different contributions from different types of evidence. One way to limit yourself to interactions is to go and find experiments here. Okay, the column with experiments sorted, and then pick, let's say, things that are above 9.9, for example, something like here. Okay, so let's select things up to here, then make this thing into select edges. Okay, so now we do have edges selected that do correspond to experimental data. You see that this is only subset in many cases. There's no experimental data for this particular protein here, whatever it is. Now, if we want to retain the selected nodes and remove everything else, the easiest way to do is to go to select edges, invert edge selection. So this way we're gonna flip who's selected, who's not. And now we just go to edit and remove selected nodes and edges. Okay, and hopefully I'm not gonna, yeah, it should be fine. Remove selected nodes and edges, and Well, I'm suspicious that those things are yellow. They might be selected. Yeah, so good. I picked off. Let's go back here. Let's go to once more experiments. Sort on experiments. Pick up as we had before things that are above point. Now we want to go from the top all the way to 0.9. Okay. Then convert selection in table to the selection in a network. Now we're gonna go select edges, invert edge selection. Now we can edit, remove selected nodes and edges. Okay. So now we're left with just 50, well, with 150 or so edges. So now what we can do is we can select and do layout once more. Let's say organic. Now we do have nodes that are orphaned. That's right. The nodes that don't connect. If we care only about interactions, then we can select those and then delete and then remove edit, remove where is it? Remove selected. Okay, and then remove those three things here. Once more, edit, remove, and then we can do layout. Let's say once more. So now we've got network that came from string that's more likely to be reporting interactions between proteins. Still, the problem is to keep track of what actually contributed to each particular edge to get to the experiments and see what's there. You have to go to the web page and then check it one by one. There's no way to go from here to information who contributed to actual interaction we've got here. All we're getting is a collection of scores for different types of information without pointers to where actual information came from. So this is one of the issues with Spring. The other one is that if you look at the node table and the way proteins are identified, you'll see that here that you do have as a node identifier concatenation of the taxon ID. 4932 is species identifier used by NCBI, by NIH or whatever taxon ID database. And then there's a dot and then a systematic gene name for in case of yeast. In case of other organisms, whatever is used as a second term might vary. So this is not consistent. You will find UniProt identifier in one of the columns, but this might throw away some of the applications, some of the apps that expect that UniProt ID is sitting here or in a name, not under in an arbitrary column. So this might throw some of the applications away. The way to solve this thing is by scripting, by saving this file, this table as a file, and saving this thing is easy. You just export table to a file, and you create a comma separated file that can be read in Excel or by a script, and then you use those things after also saving the edge table to modify the identifiers that you've got here. to be converted to Uniprot ID. So those things can be can be fixed, but there's no easy fix. It's sadly scripting. So that's second limitation on a string, as seen in a Cytoscape. Otherwise, people do use it. You're gonna see quite a few figures in papers that borrow the style that you see here. You don't need the network coming from string to use the style. The style is showing up in the styles, and where is it? Here's style. I think this is the one that we're using. We can convert to start to something else, let's say to default, and then now we're reverting back to whatever you've seen before when importing things from files. And the style, as you see here, can be used to render networks originating from string, but it can be also used to style this thing. Might require some extra tweaking, but in principle, there's no problem with borrowing the style here, except that then it's expecting additional information. So then it might require some tweaking, but in principle, it's possible to use the style with networks coming from somewhere else. Okay, so let's go back to default here. Okay, so that's what you're getting from string. As you see, applications are limited. As long as you want to get stuff out of string, there might be problems because of the identifiers that are used. Okay, so this is string. Now, much more useful is the intact app. Okay, we can do the same thing as before. We can go to our initial network, go to nodes, then select all the values, and then we can Control V, paste the identifiers here. Let's check what's in the options. There's not much. Well, max interactors per turn. Well, let's make it 100. Let's see. I think one would be enough, but let's do it this way. Let's leave everything with default. Try to search. Okay. To my knowledge, the intact app is using some custom servers run by Intact, so it's bypassing Psychic, and it's pulling in more information in a format that the app understands. And what you're going to end up with is a network with a bunch of stuff there. Let's see. Let's zoom in. You zoom in. This thing looks more informative because they are using gene names, and at least here. And on the right side, you're getting a panel that allows you to get to information about individual proteins and interactions more easily than looking through the table here and looking at values in different columns. So this is more convenient than default setting. If we select something here, then you're gonna get information about the selected node if it's there. Okay, now there's nothing there. There should be something there. Edges. Nothing. No, you're not getting anything. Nothing here. That's should be something here. So nothing about proteins, but for the edges, you're gonna get first, you're gonna get something called MI score. This is a score that's supposed to tell you how reliable the data supporting a given interaction is. Okay, and if you want to see only subsets of those subsets of edges that are more reliable, then you can use the slider to get things filtered away. Okay, according to information provided by intact score, about somewhere about 0.5, 0.6, so somewhere here is a cutoff between a cutoff above which you're getting reliable data. That's pretty decent. So that's why this thing is useful. Still, there's no clear-cut definition what it means. Well, there's a formula which tells you what's going to be retained, what not, but for one particular interaction, you'd have to check actually what's contributing to a given edge that you're looking at. You can check this thing easily because if you go under selected, let's say under selected edges information, but I've got to select something. So we'll try here. Okay. So if we select one of the edges, then there's information. What are the proteins? Okay, and then if you unfold, you're going to get a little bit more. If you want to get to evidence, you've got to switch from summary. This is just summary where you see one edge per interaction without any details displayed directly. But if you go to evidence, then you're gonna see individual edges expanded into more than one, and each edge in the evidence view corresponds to experiments. So now if we select those and look under selected, and we go under selected edges info, then you're gonna get access to information where each thing came from. So let's pick up one. Let's see here. Okay, if you look here, you're gonna have information about what is the experiment supporting, one of the experiments supporting interactions between those two proteins, and you can go, you can get interaction type, you can find where the proteins came in contact for the first time. This is equivalent of interaction host. You're gonna get information about what was the method to detect the interaction. You're gonna get information how the participants were identified. In this case, it was most likely mass spec LCMS. Okay, this is if you don't want what this thing means. As you see, each term that you see here provides a link to OLS site I mentioned yesterday. So if we click here, we should be able to get to the OLS site with information what was the meaning of the term. So this is much easier than going through the table that you're getting in a base version of Cytoscape. You can get to things this way in a more easy way. There's also the mutation version of the view, okay, and then in this case we're not going to see anything because this is a network from Saccharomyces, and probably data is there but wasn't curated. The effort is on curation of mutations in a human. So there's nothing here that we see here. But you would have edges with mutations highlighted, and then you'd be able to see in a panel on the right what are the mutations and pointers to papers reporting those. So looking through the interaction networks coming through intact is much easier than imports done in a raw cytoscape, because then you've got lookup things in a table. So this is convenient. Now, the network, as you see here, imported from Cytoscape, can be analyzed in an easy way by third-party apps because it's custom built and it's stored in a way with things in the tables that don't look that good to other applications. But what can be done is, once you let's say filter, you will go back to summary and go to filters. Let's get a little bit more of those things here. So if you decide that the network here is the one that you're happy with, then you can always extract a current network you see for analyzers, okay? And then you will have another network here. This is the one that we started with all the extra information and extra functionality that's provided by the Intact app. We can go to the exported version of network here. And this is something that's standard Cytoscape, at least it should be. We can check it here. Let's see. Well, it's not exactly standard, so I still can complain because the names are strange. Okay, so this still gonna throw applications off, sadly. So this is still doesn't help much. If we go to node table, let's see. You see the problems. They are using protein gene names for the protein name, and you're using preferred ID column to store the Unicode identifier. So this is still a problem. We can check if this is specific to yeast or is it general by trying the other network. Why not? We can go to File, import the other one we've got on hand. With File, import, and then import network from File, and I want the PRC one. Open once more here, and here, and rid of it. Okay, and then that's okay. So now back to node table, copy column values, and then back here, back to exact query. And I want to get rid of this, paste my new list, and then search the network. As you can see, something bad happened to the icons here. It's something's messed up, but I think I can live with it. Okay. Now our ubiquitin is creating problems. Clearly, clearly intact is recognizing that this might be ubiquitin because you're getting 4 options for ubiquitin in human, plus our original one. I'll keep the original one here to disambiguate the search term that we're using. And then we're gonna build a network. Okay. Okay, and here's the network now for the for our polycomb complex here. The same thing, we can filter some around 0.5 or so to make it smaller. Okay, and then we can extract for analysis here, and now we've got the simplified version here. Let's see what are the identifiers this time, and it's still the name of the gene. So once more, some applications might complain because they expect the name to be the let's say Unicode identifier. So this is the same issue as with string, where the use of the identifiers is not that convenient. Okay, so that's how you're getting data from intact, and then independent of the way we got data in either from Spring or from intact, using the apps or getting stuff directly through Psychic. Next step is typically cleaning up the network we're getting, because some things are either not useful or plain wrong. So some things should be fixed. So let's make a 5 minutes break, and then we'll continue on with cleaning up the network that we got here, to see what are the things to fix. So let's break for a 5 minutes or so. If there are questions, ask. So okay, stop, pause, Yonri, and let's continue on. Okay, so before I mess something up, I'm gonna save this whole thing as at this time. Let's call it network. Let's call it network 2. Okay. So just for the comparison sake, I'm also going to get the same network as here, but going directly to Psychic, bypassing intact. So I do have the list of proteins conveniently hidden here, so I can do once more copy column values and then paste them here, and then search. Now what we've got here is kind of okay. We can go to IMAX, okay, and then import. And then importing IMAX through Psychic should be more or less equivalent to importing data through the intact app. I mean, those two data sets should be very, very similar, so it shouldn't be that much different. Getting data through IMAX might be slower because the files that are transferred are relatively large, and then they take time to generate and parse. So that's the reason for the difference in how fast it is. intact app is using a smaller version of the files, more efficient to be sent through. So that's why it's a little bit faster. But information content should be essentially very similar. So let's file and save it once more. Except that if, and you've seen this thing before, if you when you get stuff through Psychic, then you don't have any other way of looking at the data but checking what's in the table, either for the edges or for the nodes. So that's what we're getting here. If the layout is initial is too crowded, you can always go to layout tools and then space things further away just to see what's there, or just use scroll bar. And as you see, you're getting the same type of information as before for one particular pair of protons. Let's say, let's so for one here, so for one particular pair of proteins, you're gonna get as many edges as the experiment supporting it. This is equivalent of looking at intact network here in a evidence mode. So this is the same idea. Essentially, evidence mode for the intact app is the same as, or very similar to whatever you've got here. Except here, to get to information about the nodes, you've got to check tables in intact. You've got a panel where you can look this thing up in a more easy way. Now let's see, what did we get here? Okay. If we check what's here, first let's check the proteins. Okay, if we sort on taxonomy or taxonomy ID, this is equivalent in a sense that there's one-to-one correspondence between the taxon name and taxon ID. This is similar to controlled vocabulary terms, where you've got one column that's readable by computers and standardized, and the other one is human readable. You're gonna see that there are most of the proteins coming from human, because we started with human proteins, so it's reasonable that we put in interacting partners from human, but there's more stuff. We're gonna get, we'll get various pathogens. You're gonna get information about proteins from viruses or from bacteria that are pathogenic interacting with human proteins. This is why those things are pathogenic. They interact with the host cell and mess something up. So if you're interested in those things, then you keep them. If you're not, if you want to go only after human proteins, then they've got to go. Okay, so this is the case for our RAV virus. We don't care about SARS, so it's gonna go away. You can always convert this thing to a selection in a network, and if you want to check where those things are in the network, you can use fit selected, and they should be somewhere here. So if you now zoom in, we've got those few proteins sitting somewhere here, and if we don't want them, then we go to edit, remove selected, and we've got a little bit less things. Now this is Arabidopsis. Okay, it happens that someone made an experiment which checked if some Arabidopsis protein interacts with some human protein. My guess is that this is because there's a homolog in Arabidopsis are very similar to human protein, and someone checked if those two things interact. It might be the science behind it might be interesting, but we don't care. Arabidopsis proteins don't normally interact with humans, so we can get rid of them. Okay, so let's get rid of this thing. I don't know what this thing is, chat D, but if I wanted to, I would go to NCBI taxon ID. Let's see. NCBI taxon. Okay, I will go to taxonomy page, and then I can just use the number here. This identifier in taxonomy ID is the same number as used by NCBI. Here, control V. Copy. Check here. And this is some fungus. So this is probably another case of homology, or it could be it's pathological pathogen of some sort. Either way, the name of this thing doesn't ring a bell, so we require more testing, but we care about protein interactions in human, then we can get rid of this thing. So select, and then once more edit, remove nose and edges. Now there's entire cluster of taxonomy taxons that's chemical synthesis. Those could happen in two cases. It's either small molecules, metabolites, or drugs, or things that are chemically synthesized that were tested for interaction with some proteins, or it could be chemically synthesized peptide. So we've got to be careful here, because some of those things might be peptides, might be pieces, let's say, of those histone things, like histone tails that are chemically synthesized for given experiments. So it requires testing, but typically, if identifier for the name doesn't look like Uniprot, it's something that we don't care, because it's most likely identifier of a small molecule. It could be also DNA if here if we look at the description. Here it could be a piece of double-stranded DNA that was just synthesized, just oligo or two oligos braced together. So there might be things that made sense when doing experiments, but we don't care. So essentially we can select those things, checking none of any identifiers, nothing here, so everything down to here is once more happened. Okay. Here to here, here to here. Getting messed up. Okay, so let's we have selection fine. Here to so, and then right, and I okay, and now select from same drill as before, from selected, and then edit and remove. Okay, what else do we have? We've got Drosophila here. We've got some strange thing here. This is a virus. You see identifier here looks like the identifiers I mentioned yesterday. It's a UniProt ID dash a region of likely protein. So this is probably another case of a virus. Okay, couple of proteins that are probably homologs in Drosophila and so forth. I'm gonna remove all the things here, same drill as before, and now select from okay, so everything before human is gone, and then we go down here, and there's more interesting things. Now, so another virus, apparently some mysterious I33, which I don't know what it is, but doesn't look like a human, so we'll get rid of it. And then we not too surprisingly get quite a few proteins from mice. This is not surprising because looking at mixed interactions between mouse homolog of a human protein and human proteins is relatively common experimentally. Okay, so there's a mouse here. Then rat is the same case. There are a few these genes here. What this thing is, I don't know, but one can always check by using the taxon ID and checking on a taxonomy page, and then you're getting these proteins, same reason, probably interaction between homologs. Out of 2,000 or 2,200 or so experiments, we do have in our case about probably a couple hundred or so things that are not human. I mean, out of 900 proteins we're getting, let's see how many. Here, do select, and then whatever is selected in terms of nodes and edges, there's a counter here telling you that we've got 80 proteins selected. So we're losing about 10% of the proteins, including whatever we had before. If one is really paying attention to details, one might try to convert some of those interactions to something which is usable, which essentially means looking for orthologs in human corresponding to these proteins and then substituting the identifiers. This can't be done by clicking. It's writing script. So this is more complicated thing, but losses are not that large. If interaction between yeast homolog of whatever human protein is relevant, someone probably did experiment with actual human protein. So probably the homolog to this particular protein in yeast is present somewhere in the network. So so the loss might be not that great. So anyway, we're gonna remove this thing. So just edit, remove selected nodes and edges. Okay, so now we ended up with a network with only human proteins. So this is something useful. Okay, and that's the part dealing with cleanup of the problem. There's one more thing. Now, if we sort by name or any column that is using UniProt IDs, you're gonna notice that. Oh, okay. So we dealt with species. Okay. So second thing to deal with is going to be the type of the molecule. Okay. There's gonna be a column called interactor type. Okay. If we sort here, then we will see most of the intractor types reported as protein. If we care about PPIs, then this is what we're interested in. But there might be more, so it's worth checking. Okay, and if we check after sorting, we're getting few positions reporting few intractors being actually pieces of DNA, either double or single stranded. In this case, we've got either unqualified or double-stranded DNA. We might have a case of gene. Those are probably experiments that are chromatin immunoprecipitations or chip or whatever. So we're gonna get rid of this thing because those are not PPIs. The same as before, we select the block of intractors we don't want to, and we convert this thing into selected nodes, and then we do edit and remove. Okay, and then what's at the other end? We're getting ribosomal RNA single-stranded DNA stable complex. This is probably interaction between protein and the complex of two or more proteins, which we can't deal with. So this is gonna go also away once more. Select from the nodes, edit, remove nodes, and add this. So now we're left with proteins only. That's right, everything in this column says protein. So this is second step. And then third step is to sort it on uniproduct session or any field where it's used. So it could be either here, it could be named, those two things should be similar. And then if you scroll, and look carefully at the identifiers, you'll start to see either identifiers missing, which probably means something strange about particular intractor. So if needed, those things can be checked for what those things actually are. The way to find out what each of these things is to take the identifier from the name, if it's not UniProt ID, make a copy of it, and then you go to intact database. Okay, intact portal. Okay, hopefully it's alive. I don't know. Is there still UK? Oh, yes, it's still there. Okay, and then you paste the identifier here, and you should be able to find out what this thing is. Okay, so it might be guessing by name. This might be a good, what is it? Plus F molecule. This might be, it's my guess, this might be a fusion of two proteins in some cancer cells. This might be one thing. It would require further investigation what this thing actually is. But the point is, you can get anything that you see as coming from IMAX with identifiers that look like EBI dash something. Those are identifiers in intact, so those things can be checked on the intact page. But for now, we can get rid of those. Okay, same drill as before, selected and edit and remove. Okay, so then we're back to identifiers. We're sorted on identifiers. Okay. So what next? Okay, let's keep going. Is there anything looking different than UniProt IDs? Nothing so far. So now we're limited to UniProt identifiers of some sort, but every now and then you see those dash twos or dash somethings here. And then if they are alone with no corresponding undashed version of accession, then the situation is relatively simple. There was just experiment that was using one particular, most like clone splice form of a given protein, nothing else in data set. So this is more or less fine. The worst situation is when you do see things, and there are things like what I'm trying to find. Let's see. So here we've got two different splice forms of the same protein. That's right. The first part of the name is identical between those two, but there are two different splice forms, which means that we're gonna have, instead of two proteins interacting, instead of one protein with a bunch of interacting partners there, so instead of our protein and binding, so we're gonna have two variants of this thing. Like this is gonna be -1, this is gonna be -8, and then you're gonna have interaction here with experiment done with -1 protein, and then you might have experiments done with -8 version for other proteins, and for some of the partners, some experiment might be done with this thing, and some experiments might be done with this thing. Okay, so the network starts to look different. You're splitting one of the proteins into two or more things. More common thing is that you've got dashed thing and then another version that's undashed. So then you've got something without dash. That's P1, 2, 3, 4, 5, and then you've got P1, 2, 3, 4, 5 -1, and then another one is P1, 2, 3, 4, 5 -8 or something like that. And then the resulting network is messed up because instead of 1, you're ending up with 3, and some of the interacting partners are reported to bind to this, some are reported to bind to this, some are reported to bind to both. It sort of matters because then if you try to see how similar from this guy, okay, and then if or between this one, if this thing is collapsed into one protein, it's one edge to go from here to here, another one to go from here to here. But if those two things are split, there's no direct connection between those two, then you've got to go here and here. So instead of being 2 edges away, here like 1, 2, 3, 4 inches away. It might matter if functional based on the length of that. And also those things are to be avoided. And we've got the case like here with two dash things. There's more of those. I've seen one. Oh yeah, here is the case. Here, 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 and here. Let's see how it looks like. Okay. So you see, so let's get those things out somewhere. So making selection of several nodes is convenient because then if we want to move them away, you can select all of them. And then if you drag it away, you can isolate them from the rest. So you see, so those three things here are essentially the same or very similar yeah. Here, select, and here. So here's what we've got is we do have this generic identifier, and this is most commonly reported form. This is because if in most of the experiments, as you've seen yesterday, it's relatively hard to identify which splice form you're dealing with. So most of the reports are gonna point to just generic identifier. There are 1, 2, 3, 4 experiments where either this splice form or that splice form was either identified with enough accuracy to specify which one you're dealing with, or it could be that those are 4 experiments where either this variant or that variant of the protein was cloned, and then you know which splice form you're dealing with. So you've got those four things hanging off. And if you and there are 2 strategies. Either you ignore the splice forms unless you really care about this particular splice form for your particular system, it might matter. So then if you're working on this protein itself and you care which splice forms interact with what proteins, then you might care. In most of the cases, if you just care about generic topology of the network and you want to see if the proteins you're interested in are within one region of the network, not with the other, then you can just keep those things away. You're not gonna lose that much because you see the difference, how many interactions experiments reported for the generic identifier versus dashed version. So you're not gonna lose that much. And if it's somewhere in a network far away or not that close to wherever your proteins are, you don't care. But if this is your protein, you probably care. So if you want to fix this thing, it's possible, it's tedious, but it's possible. The way to do it would be to take, well, first file and save this thing before I mess up something. Okay. The way to do it would be to take the net, would be to take and save both edge table and the node table. So those can be exported to a file. Okay, I'm gonna change this thing to here. Just want to have an edge, make it edge, H1. Okay, let's save it here. Okay, and then you're gonna end up with a file here. This is my H1 file that can be opened in Excel. This is exactly the table we had before. And then there will be mutation author complex. There will be a column with the, let's see, where's our Unicode identifier column? Well, no, this doesn't have direction. Oh, here. Okay, so there's gonna be a column using UniProt identifier, then brackets, and then another UniProt identifier. This is a name of the interaction. Okay, and it's got the starting node identifier, which is UniProt ID, ending node identifier, which is also UniProt ID, and as an interaction type, which normally would be PP, IMAX imports are using the identifier of the experiment. So those center things vary between experiments, but they might be padded with same proteins on the sides. Like you've got here, you've got the first two interactions between the same protein, the 833 and N18, both here and in the second line. But the interaction type, which is a proxy for intact data, for the experiment ID is different. So there are two experiments demonstrating interaction between those two proteins. So now to solve our dash problem would be to find entries in the name column. And if there are entries with dashes, which might be hard to find, but let's say something like here, then it would be to remove those dash ones, remove all the dashes from the name column. It means that we would be converting interaction. Let's say 1, 2, 3, 4, something. This would end up as interaction with 1, 2, 3, 4. Here. So, as written here, those two interactions would be like and if you remove the dashes here, those two things would be merged into one. So now you would have just one interaction with two supporting experiments. So literally you're going to combine all the nodes into one. Okay, and then once this thing is imported in as a file, then you're going to recreate the network you started with, but you're going to collapse all the splice forms into one entry. But then you're going to have a mismatch for the protein table, because when this thing is imported back, you're going to create the network topology, things that are going to be connected. You're going to get annotation in an edge table, but you're not going to have annotation for the proteins because this thing is lost. So to get annotation for the proteins, you got to do the same thing with the node table. You'd have to save this thing and then collapse the identifiers for the nodes by removing the dashes. And then you import this file to annotate the nodes. And then you're gonna have corrected version of the network with all the ambiguities, uncertainties about the splice forms collapsed and removed. So this is doable in Excel. It's relatively simple. I just don't know, but by doing search and replace in Excel, it's doable without any scripting whatsoever. So those things can be fixed. So this is something that after initial import can be done to fix the network. So this is second step. And then the third step is to go through information about experiments. So instead of looking at the node table, we're gonna go to edge table. And here now we're gonna go under interaction types and sort on interaction types. Okay. Now, what do we see here? Well, if we scroll, we're getting a bunch of associations. Then we're going to get, what's the next one? Then association is, as I mentioned before, the mislabeled physical. Associations reported for pairwise interactions must have come from spoke expansion of affinity complexes, which were originally annotated as associations. And IMAX data is coming in as expanded for the associations for the QIPs and things like that. But sadly, the interaction type is wrong. It should be, instead of association, it should read physical. So this can be fixed once more as with the identifiers for the splice forms by getting this file into Excel and then substituting interaction type if there's association converted to physical association, and then you correct this problem. Or just ignore it, just when seeing association in the network, assume it's physical. Okay, it's depending if you want to use this thing for processing somewhere later on, or just for looking. Okay, more serious problem is colocalizations reported here, because those experiments do not demonstrate that things interact. They demonstrate that two things are in lysosome, or two things are in nucleus, or two things were when immunostained were observed in the same area within the cell. But this thing should happen for things that interact, but the reverse is not always true. Things that colocalize do not have to interact at all. So those things, if you're looking after PPI, should be removed. So the same drill as before, you convert this thing to select edges from the rows and send this thing out, remove things. Okay, what else do we have here? Associations. Okay, so then covalent. So covalent binding is stronger. This is proper physical thing. Ubiquitination, deubiquitination. This is also fine. If you've got two proteins, one ubiquitinated, mixed, and ubiquitin comes off, probably the other protein was an enzyme that was getting the ubiquitin off the ubiquitinated protein. So this is proper direct interaction. So this is stronger evidence, stronger than direct. So we're fine. Then we've got direct things. This is also fine. Okay. As you can see, by the time I went through associations and got to direct interactions, I'm 2/3 or a little bit more than halfway through the list of interactions. So it tells you that half of the data that's there are just physical things based on expansion of QIPs and pull downs. It tells you roughly, gets you roughly an idea what's in the data and what experiments interaction data is based on. Okay, so then we go to direct things, and then once more, then we've got enzymatic reactions, which are also stronger than direct, in a sense that it means that one enzyme was substrate, the other one protein was substrate, the other one was enzyme. So they did have to come in direct contact. So those are evidences. Those are interactions that are actually corresponding to physical interactions between proteins. We've got phosphorylation, once more kinase and kinase substrate there. So that's fine. Then we've got physical associations, and then what else do we have? Physical. Okay, good. After physical, there's another interaction type, another term, proximity. Those are experiments that demonstrate that proteins are close together. It's a more specific version of colocalization. The idea is, and this is recent 5 or so years, those are experiments where you do tag a protein with an enzyme that generated activating biotin, as far as I remember, which when added to the sample is gonna get activated, fly off the tag, and then attach randomly to things around the protein. So the idea is that you have the protein on the tag that's enzymatically active and generates an activated version of biotin. Okay, and then this thing flies off the tag, and if there's another protein nearby, this thing is gonna attach itself to the neighbor. Okay, and this tells you that you've got those two proteins nearby, one that's doctored. It doesn't tell you that those two things are in physical contact with this thing. It just happen to be somewhere nearby. So it is a specific version of colocalization. So those things probably should be removed if you don't care about colocalizations, just about physical interactions between proteins. So this is something to get rid of. Okay, so then once more you select, edit, and remove. Okay, what else do we have? Physical, so we're physical ubiquitination is fine. This is opposite of deubiquitination. Once more, protein plus enzyme that's ubiquitinating things or something like this. So this is fine. So now we can clone ourselves. This thing, new network, clone current network. And here we do have archived version of network that was cleaned up. So this is something that's more reasonable in terms of protein identifiers, in terms of molecule types, and in terms of interaction types. So this is the current status of the network. Now, is there anything else to fix? Let's see. Yes, there is. Okay, so now the problem at this moment is if we do once more some sort of organic layout, and the problem is that we still do see multiple edges connecting some of the proteins. How to find those things? To find a good example. Well, this is sort of annoying if you're too far, then the edges are collapsing, so you can't tell. Don't want to collapse. Oh, okay, here is a good case. Okay, so we still do have situation like here where we've got two proteins. We can do it this way. We can select those two things. Okay, so now we do have two proteins, okay, with two experiments demonstrating that they interact. Okay, if we select those things in the edge table, those two things will show up as the only two things visible. And then when you look here, the interaction types for individual experiments might be different, because one experiment demonstrates that it's a physical association, the other one might demonstrate something different. Okay, in this case, it's the same because association probably came from expansion of the quiet piece or things like that. So this is actually physical. So here there's no contact, no conflict. But in other cases, you're gonna see pairs of proteins connected by edges with different types that are really different. You're gonna see things that are direct and physical and associations and whatever. And you'd like to know which one to retain, right? If you collapse the edges, okay? Collapsing the edges is not a problem because this is what we did before, right? We can easily go to edit, remove duplicate edges, and Cytoscape is gonna retain one of those two things, but there's no clear order which edges are retained, which are removed based on interaction type. This is completely random, and I think it corresponds to the order of adding those edges to the initial network. So the first one is gonna be kept, and the next things are gonna be removed. In the first time happen to be direct, then it's fine. But if the first one was association and direct was the last one, the information that this is actually direct, or the information this is actually direct interaction is gonna be lost. So the proper way and clean way to collapse the multiple edges is sadly by scripting or with painstakingly in Excel. And one way, well, that there's probably a way to do it in Excel in a very roundabout way. What you do is you save the interaction network as a table, okay? Then, well, okay. It's way too roundabout way to bother. Okay, it's doable, but it's multiple steps. So probably the easiest way to do scripting, to play with Python or whatever to process the files exported from Cytoscape as tables and do corrections there. The simple thing is is just removing duplicate edges, okay, from, let's say, this last one. So I want to do it from my guy here. This is IMAX 5, yeah, okay. We will ignore edge direction because things like bite prey and prey bite might be in the opposite directions reported in a data set. So we want to collapse everything, no matter what's the direction. So ignore. But we'll create an edge table with number of duplicated edges. So at least we know which edges were collapsed, which are just supported by a single experiment. So we can create the table here, and then we do OK. Okay, we lost 800 or so duplicated experiments, and this is reflected on the count of edges between the network we started. So 1800, we ended up with nearly 1000 or so edges, but those are unique to pairs of proteins. So this is our duplicated thing. It should go, should be gone by now. Oh no, lost it here, but duplications got lost. So now we've got things connected by single lines. And then if you check at the very end of the edge table down here, there's gonna be a number of underlying edges. So this is the number of experiments that contributed to a given edge. If we sort it here, then here, so there's one interaction supported by 26 experiments, few interactions supported by 10 experiments. The vast majority is hanging off one experiment somewhere here. So this is how many of those. Okay, we select things here. So we select edges from select. So about 2/3 of edges, I'm guessing, getting this thing from total count of 900 something for entire network versus 600 something for selected nodes with the one supporting experiment. So about 2/3 of the experiment edges interaction is supported by a single experiment. So that's also typical. Most of interactions are supported by very few, one or maybe two experiments. Only small set is supported by quite a lot. It tells you something about how research is done, that there are proteins that are more interesting than others. And it also tells you that some of the interactions are more reliable in terms of being supported by many diverse experiments, versus some of them are hanging off some single experiment. And oftentimes, if you look at those things that are supported by single experiments, so let's look at those. Let's see. Look at those here. Select from row, and then one of the columns in the edge table will refer to a paper where the interaction experiment was generated and described. Okay, and if we sort on the author, you're gonna see that for those experiments supported by one, for those edges supported by one experiment, you're gonna see very few papers listing a lot of experiments. That's right. You're getting this other whatever Aleraso et al. Reporting how many experiments? Let's see. And select. Let's and select from rows. Reporting 17 experiments. There are worse offenders. You're gonna find papers reporting 200, 300, 500 interactions all at once. Those are those high throughput papers which did a high throughput screening of interactions. They reported possibly those would be either for all their papers, this would be to hybrid. It would be some other complementation assay, or those could be affinity purification plus mass spec done in a high throughput way. And those experiments were not followed up by anything else because there's no other experiments reported for them. So those are the most suspect in terms of being real. So if you want to have a very highly reliable network with not much noise coming in from high throughput data sets, you might consider removing all of those, because then, well, you're gonna lose quite a few interactions, but the gain is that whatever you've got there left is supported by at least 2 experiments. So there are at least 2 independent pieces of evidence suggesting that the interaction is correct. So if you want to go and improve the quality of the network, you might consider removing those experiments coming from papers reporting, let's say, above 100 experiments. This roughly corresponds to whatever you do when sliding or setting cutoffs for the reliability score or quality score. The first thing that's going to remove when you start to go through quality control done by some sort of algorithm is you're going to remove those high throughput things. So this is equivalent of the sliders, and you can do it in a controlled way. If you read those individual papers, you might have your own opinion which data sets are better than the others. You might decide on your own which to keep, which to remove. So that's last step in the cleanup of the imported data. So that's how it's done, and it can be done for, as I was playing here with the data imported directly from Cytoscape, but the same thing can be done on the data set that was exported after getting stuff from Intact App. And so the same thing can be done in combination with setting up a cutoff for the quality control as done by the Intact App. So by using here the filters here, species filters here. Where are we? Wait, where did I lose it? I lost my. Where did I lose my slider with the quality? Huh? I lost it somewhere. I lost some legend, no notes. Oh, the sitting under edges. Okay, here under edges, you've got filters and you can combine filtering here with doing manual cleanup after that, or you can apply filters of various sorts within the app. Some of those will be equivalent of what I was doing by looking at the table. So those things might simplify things a little bit. In particular, it might simplify fishing out the correct. No, it's not gonna fix. Well, it's gonna fix some of the problems with interaction type because you can remove some of them here. But when combining edges, it's still gonna be the problem. So for as far as I know, whatever is coming out here. Let's see. I check here for the summary, and then if I check the edge table, how the type looks like. The summary is if I remember correctly, the interaction, it's not. Let's see, check this one, ASF, and then interaction. It's interaction. Yeah, there's nothing showing up here. And then if we expand this thing, yeah, so you're getting information about interaction type in the evidence view, but if you do summary, apparently this information is gone. So you can't have a summary exported because you're gonna miss the information about the interaction type. I think so. So some things can be made easier within the intact app, but not everything. So somewhere there's gonna be cleanup, playing with the tables out there. So that's. But there's nothing wrong with combining whatever you can get out of the app plus manual fixes. So that's how the cleanup looks up. Okay, so let's break once more, and then we'll try to use this cleaned up network or the simpler one from the East cytochrome complex to see how to work with this thing with the bingo and looking at the functional enrichment and like that. So let's break for a moment. So okay, so now I'll save this thing just in case something comes up. And now, okay, so one thing to look on the string side. Let's see, do I have it somewhere here? No, so let's go back to strings. Okay, and let's go here, and then let's pull a random protein from our cytochrome things. Okay, let's pull out one of the proteins from cytochrome and get it here. And that's the one. And cut and paste doesn't work. Copy. Oh, well, no, this is not. This is something random. back. Okay. Paste. Okay, here's the identifier. This is Saccharomyces search. Okay, so one thing that the string website reports is, if I think you go here, it's gonna tell you for the network that you see here, information about the gene ontology terms of the functional annotation of the proteins, how often terms are used for this set of proteins as compared to everything else in the list. The idea is that if a set of proteins you've got here corresponds to some sort of a functional unit, let's say a group of proteins that are parts of a complex function in one specific way, then most of those, if not all of those proteins, will be annotated with the same term, which for a random collection of a dozen of proteins is relatively unlikely to happen by chance. So then you can calculate what's the probability of getting a term used for all the proteins here on random, and this is roughly what you see here. You're gonna see I started with a protein that's a part of the respiratory chain complex 3. So it's not too surprising that you're gonna get terms corresponding to electron transport, to mitochondrial ATP synthesis, and so forth, showing up more often than on random, and you're getting pretty high or pretty low, depending what you think about p-values for those terms. Okay, so you can get this thing in a very easy way from string. The problem is that the enrichment that you're getting in string, the numbers that you're getting here, are relative to entire yeast genome, or whatever genome you're working with. This is not exactly correct. The reason is that if you imagine a situation where you're looking, let's say, for proteins in some sort of cells that you treated in some way, you found proteins that got the expression level going up, a subset of proteins, those cells is gonna go up. Your reference is not entire genome and not the levels of expression in an average cell, but it's something that's in your test tube, one particular cell line. This cell line will have some biases on its own, because it's gonna be a liver cell, or it's gonna be a some immune system cell or whatever, and those cells do have the basic levels of expression of things different than the average. So this is one issue, and second issue is not all the proteins can be detected in a given experiment. So this is gonna introduce another bias where you've got a subset of proteins that are detectable in your cells, and this subset might already correspond to some sort of enrichment of functional annotation terms. So the string is completely ignoring that, and it's starting from everything in a genome. And then if you feed string with the set of proteins that you can detect in your experiments, even before testing for whatever condition changes in whatever conditions, you might end up with biases and string telling you that, you know, the whatever functional term is enriched. So for initial screening, it's probably fine, but in reality, something more fancy should be used, and that's where among others, the bingo comes in. Okay, this is another app that you start if you imported it by going to bingo here. And then there's quite a few options here. Okay, first we do check get cluster from the network, and to use it we've got to select things here. I'm gonna select everything because this is my initial set of proteins that I'm starting with. So I'll select everything. I'll go back to bingo here. I'll have to give it a name. So let's say that one. Okay. Now, there are two ways of testing for enrichment. They differ in speed. The hypergeometric test is I think computationally more intensive than binomial. So for large networks of thousands and thousands of proteins, binomial might get you results faster. For those tiny things, we're dealing with hypergeometric is fine. Okay, and they should be consistent in terms of results. So it might be worth testing both. Okay, now we do have to make a correction for multiple testing, because each enrichment analysis is a test repeated for every term. So if we run 1,000 tests with and we set up cutoff for p-values at let's say 5% confidence level, if we don't do correction for testing 1,000 times, you're gonna end up with 5% of 1,000 things hitting our p-value of whatever is corresponding to 5%, just because that's how p-values work. So if we've got to do correction for that, which essentially means making the p-value criterion more strict. There are two ways to do this thing, either Bonferroni or Benjamini. Bonferroni is more restrictive. It's more stringent. The Benjamin is less restrictive. So if there's nothing coming with Bonferroni, then you can try the other one. Okay, significance level, people picked 5%. So let's steal here. We are looking for overrepresented categories after correction. So we're looking for enrichment of the terms. It can be done, and it can be either before or after correction. So we can do after corrections. There might be something. No, there's only overrepresented. I hope there's underrepresentation. Nothing here. Okay, so this is fine. And now I'm gonna use whole annotation as a reference set. This is equivalent to what string does. In reality, you would do custom, either custom and provide the file with whatever information you need, or you would use network as a reference set, and you would make network based on your background, your initial things that you can measure. So if you've got your experiments where you can identify, let's say, 1,000 proteins or 5,000 genes, you would make a network that contains all of that. And this would be your reference set for the enrichment. And this way you're bypassing the problem that there's a bias in a set of proteins that you were observing. And then you're looking at the change in conditions where it can only affect the set you're starting with, not entire genome. So this is correction for cell line or tissue type or whatever. So this is something you can do here, but you cannot do in string. So And if this thing is not done, then you can get funny things happening here. And then we do have a choice of different sets of annotation terms. Typically, what's useful is gene ontology biological process. So this is gonna be information about the pathways or modules or something with meaning in biology. Another thing that might be useful is cellular component. If you, this might also provide you information where the proteins are localized, and this might tell you something about what's going on. But typical thing is geobiological process to pick. You do have to specify the species, okay, and Saccharomyces, so that this is okay. Now, the gene ontology terms are taken essentially from UniProt. Each of those things is coming with so-called evidence codes, which specifies how a given gene ontology term got associated with a given protein. Some of those things, as I've shown, I think yesterday, do come from experiments performed on a given particular protein. Some of those things are coming by transferring from a homolog or by some other computational mean. And then if you don't want to use them, then you can specify those evidence codes here. You can find those things when checking gene ontology website and see what those things are, and you can list those things here, and then you're gonna use more reliable gene ontology terms to look at. So this is another place where quality control can be done. I'm gonna skip this thing here, but I'm gonna say that I want to save data in let's say documents. That's fine. Okay. No, let's go to desktop W22 here. Okay. And then you start bingo. And nothing happened. Why is that? Because something is wrong. Let's see, go back to here. Let's try it once more. Something got messed up. Tools, apps, this is the to get from network hyperjump overrepresentation. Oh, here the overrepresentation versus underrepresentation is here. Okay, biosets should be fine. Biological process. Okay, so let's save. And Saccharomyces. Browse. We want to go to desktop here. If you don't want to repeat the selections over and over again, you can save settings as default. It should remember the choices. And why doesn't work? Start. This does not any wrong type of gene identifier. This is typical problem with okay. Bingo is very picky about identifiers. This is this thing I mentioned about different databases, different apps using different identifiers for the protein. So if completely nothing nothing is coming out, it's probably problem with identifiers. So let's, for a quick test, try the other thing. Let's try, because I don't have, this is the wrong thing. Where's my cytochrome thing? Because this is PRC. Oh, no wonder. This is the wrong network. Okay, let's try this one here. Tools, tools, did I screen bingo here? Okay, set 3. Hyper, whatever, G pathology, compromises. Okay, let's try now. Okay, and I have switch. Okay, start. Oh, here we go. Okay, so here how it should look like. Okay, so Bingo is gonna return you a table with information what it found. This thing can be found in directory that you specify when running it. So this thing is accessible. In a W22 directory in my desktop. So there should be set 3 here. This is no, I want to open it with Notepad and then file, open, set 3. So this is essentially the same table that you've got here, just as a text file. So this can be read into Excel. So you've got it here, okay? And then what you're getting is a list of gene ontology terms with identifier and description of what the term is actually. You're gonna get p-values, the raw values, and corrected after this Bonferroni or whatever was picked up for correction. Okay, and then information about how often this thing shows up in a cluster, and then information which genes or proteins are mentioned there. If you want to select those things in the network, you can put checkbox at 1 or 2 or more terms, and then select nodes. And then what we should see is nodes selected in our starting network. Well, let's see, because we had something selected here. So go back here, select, let's say, those 3 things, select now. And it should be here. Yeah. Oh yeah. Okay. Yeah, that's okay. Yeah, I'm picking up the wrong thing. So this way you can track which terms are associated with which protein. So this is what you can get out of Bingo. Now, what's interesting is to take this initial set of proteins, okay, add interacting partners, and then rerun Bingo analysis on things that are not the starting proteins, but the neighbors. And then you're gonna see if you can guess what was the initial function of the set of proteins that you started this thing with, by looking at annotation of the proteins that this initial set of proteins is interacting with. This is so-called, referred to as a guild by association, proteins that interact with my protein. What they do is similar to what my protein does. So if I don't know what's the function of my protein, but I know that it's interacting with a bunch of proteins within let's say respiratory chain, my protein is likely a part of respiratory chain itself. So this is the rationale behind all this enrichment analysis and using networks to look at enrichment of the terms that are used to annotate proteins interacting with proteins in my set, which I might not have annotated at all. So this is one of the application of networks combined with enrichment analysis and Cytoscape plus bingo lets you do it in relatively straightforward way. So that's how bingo works. And it once more it's partially paying partially playing with this thing, checking how things work. It's a lot of what you've seen here. It does require just practice to do things. So that's the bingo part, and I think this probably wraps up the entire class, unless you've got any other questions and you want to know more, then keep asking. Any questions? No questions. Okay. So that's about it. For those people who take this thing for credit, I'll post the quiz over the weekend. It's essentially going to be based on yesterday on interaction types, interaction costs, things like that, because I think this is something which is universal. It's not some detail of how to click on in Cytoscape to do something. It's something which should stay. It should be useful for longer time. So that's what I tend to put into those quizzes. Plus, there's gonna be some sort of assignment based on whatever you've seen probably today. Something like a start from a network at interacting partners using either Spring or and then annotate the resulting network, or maybe run bingo to something like that. But it shouldn't take more than a couple of hours of clicking. So it shouldn't be that hard. And if there are any questions about answers or whatever to quizzes or assignments, then feel free to ask. It's not me who's requiring those things. I am very happy to help and make this thing work. So it's just. And the same thing, if you've got questions later on about Cytoscape and you want some help, whatever, feel free to email me. I might have answers, I might not have answers. At least I can try. So hopefully overall it was useful. And then I've got, you've got to evaluate me, darn it. Let's see, there's one more thing. He told me I'm supposed to put this thing. I mean, let's see. I have you've got to sign this piece of paper. So this is one thing. Okay. And then there's supposed to be discount general number Eloy. Oh, to protect, allow. No, this is not it here. Oh yeah, here. So you're supposed to get this thing and do something with it, whatever it is. So whatever. So otherwise, thank you very much for participation and showing up here. Hopefully it was useful, at least marginally. So I mean, there's another layer of this business that involves scripting, because a lot of Cytoscape is limited. whatever you can do it. And then the other limitation is it's very hard to keep track of what you did. With any GUI based approach, it's you'd have to dutifully either write down every click or have a webcam watching you, and so that you can replay. So from this point of view, things that are done by writing scripts are reproducible because you can always do things once more, rerun the script. So with Cytoscape, for sure, one thing to do all the time is when you get the network in from wherever, make sure you save it. Once you get the draw data, before doing any playing whatsoever, save this thing, store it in a safe place, because then you'd be able to reproduce your procedure once more. If you don't, from day to day, whatever is coming over network might vary. And you're gonna get different things, and saying that I've downloaded interaction network from intact, it's completely meaningless, because it might depend on literally difference of 2-3 hours. You're gonna see different things. So thing to make sure is, once we import stuff, save it and keep it for an archive. Keep it as an archive, just so that you can tell people that, oh, I started from this network, not from. So okay, otherwise, thanks a lot. Okay, bye. I'll send. You mean the assignment? I don't. Oh, yes, I think it will. I think stop recording. Yes.